I, Garjan, a second year undergraduate student currently pursuing my degree in Bachelor's of Chemistry, host of the today's event, it gives me immense pleasure to welcome you all on the behalf of Department of Chemistry, Hansraj College, who has clubbed all of us for the very first Indo-Brazilian seminar and scientific interaction on chemistry-biology interface. Did you know that Brazil and India share a long history of scientific collaboration? From biodiversity to sustainable development, our countries have been at very forefront of innovative research. Today marks a significant milestone as we gather here to explore the intersection of chemistry and biology. But before we dive deeper, I would like to take a moment to express our deepest gratitude to our principal, Professor Dr. Rama ma'am. Your vision and support empower us to explore the boundaries of knowledge and foster our academic growth and exploration. And also gratitude to our conveners for their support, which has been very instrumental for making this event a reality. Please give a huge round of applause. So let us begin with a tradition that symbolizes the illumination of knowledge, the lamp lighting ceremony, which signifies the ushering of light, wisdom, and prosperity. I would like to invite our guest and all the faculty members present here to join us on the stage for the lamp lighting ceremony. Thank you, sir and ma'am. Now, it's time to honor our esteemed speakers who have graciously agreed to share their insights and experiences with us today. So, I would like to invite Dr. Himanshu ma'am to present the felicitation to Dr. Lindomar Premier. Give a huge round of applause for our very first guest speaker.
A very good morning, all of you. My colleagues from Brazil, Dr. Lindo-Pena, Professor Ronaldo, Dr. Himanshu Kaushik, and Dr. Aparna Bansal. This is really a great privilege for all of us to host our guys. And since last almost 2 weeks, we have been celebrating them each moment, scientific come cultural both. It's again very interesting for me to tell that this collaboration was started in 2019, and from that time onwards we have been discussing, meeting online. I had been there once 2019, and they had been here in last conference, and I'm really glad to tell that we won a BRICS grant jointly. That has become a really ladder for scientific interaction and also the cultural exchanges. Because both the countries, when I visited there, I never felt that I'm in a foreign country. Believe me. This is something that I realized. Everyone was so humble and so cooperative, so responsive that 15 days just flew. I did not realize. So because we have the cultural similarities, we both have very good, both the countries' people, uh, they are respectful for each and every person, and they respect the humanity, and they love each other. So that's really very important. So not only scientific thing, they are sometimes byproducts. If you have a good bonding, good understanding, good interactive attitude, surely scientific things will come, because basically we are scientists. So that has to come. So this is something really very good, and also today our department, we are discussing something very interesting, that one of our student and the colleague Dr. Mansi Verma will be visiting them in month of June. The basic idea is to get some training program, and the students from their lab also will visit, I hope soon. And first, the thing is that we both the institution will have good cooperation, good understanding, and scientific exchange program. It's really glad that we could publish in last few years more than 7 publications, looks like, together, and that's a great thing. It takes time to propagate things, and to shape up. Propagate to ho jati hai, but to shape up them in a scientific, in a passionate way. So it's really very interesting, and I could see both of them. They are so passionate and so humble guys that it's really, we all are honored to host them. And I wish that this collaboration moves on. Not only 2 guys from our college or the institution, but also maybe a lot of people must visit. It's a beautiful place there, and also our next conference is being organized. I think for that you need to start accumulating, saving some money. So it's really very interesting, and thank you so much for me to speak something about our collaboration. Thank you so much.
Thank you so much, sir. Today marks a significant milestone as we gather here to explore the intersection of chemistry and biology. From the elegant structures of DNA to the fascinating mechanism of enzymatic reactions, we will uncover the mysteries of chemistry and biology, where the building blocks of life meet the forces of nature, the 2 disciplines that hold the key to unlocking groundbreaking discoveries. So get ready to be inspired, challenged, and enlightened. So without further ado, let's dive into this enriching session. And I would like to call Dr. Aparna Bansal ma'am to kindly introduce our first speaker.
Good afternoon, everyone. It is my great pleasure to introduce one of the eminent scientists, Professor Lindomar Pena, present among us in this first Indo-Brazilian seminar. Professor Lindomar did his PhD from the University of Maryland between 2018 to 2011. He also holds TBM degree, that is Doctor of Veterinary Medicine. He worked as a postgraduate researcher in Island Animal Disease Center and also as a visiting researcher at the National Animal Disease Center. Presently, he is working as a principal investigator at the Oswaldo Cruz Foundation, Brazil, a largest biomedical research institution in Latin America. Professor Lindomar has a broad background in virology with specific training and expertise in reverse genetics, viral epidemiology, animal models, pathogenesis studies, and development of diagnostic kits, vaccines, and antivirals. His research studies include viruses that impact human and animal health both. His research group is carrying out the research on arbovirus like Zika, dengue virus, chikungunya, influenza, etc. His teaching areas include biosciences and biotechnology applied to health, genetics, and drug development and technological innovation. Professor Lindomar has established several national and international collaboration with NIH, University of Georgia, the University of Toronto, Canada, University of Glasgow, Scotland, and with great pride, I would say, with University of Delhi, as Dr. Rathi has just said. And he has several patents and more than 90 research papers in International Journal of his credit. Without taking much time, I would like to invite Professor Lindemann for his talk.
Good morning, all. Namaste. First, I'd like to thank Dr. Parna for that very kind introduction. I'd like to thank you, the organizing committee, especially Dr. Ratti, for having us. We have the same feeling here, Hati. When you come to India, it's our second time. We feel at home. Although our countries are so different, right? The language, the culture, the food, the religion, which are all we love. But I think we share something very common, our human nature. Indian people are very warm, so we feel at home. Today I'm very happy, but I have double feeling because tomorrow is our last day, so I'm a little sad that we have to leave India, right? But we'll be back. Today I'll talk to you about some of the research that we have been doing in our lab at Fiocruz. I'm located at Recife, which in English means reef, because the city, it has these reefy walls along the shore. It's a very beautiful city, the northeast coast of Brazil. It's a city with 3 million people. And the research that I'm going to talk about is about the triple arbovirus epidemics that we have been facing in Brazil. The same thing has happened in India, right? Zika, dengue, chikungunya. These triple arboviruses, which means the viruses that are transmitted by mosquitoes. These are lesions of chikungunya and Zika. They are not distinguishable from each other. Only the physician cannot tell by just examining the patient if it is Zika, dengue, or chikungunya. Only the laboratory can tell with precision, right? There's some clinical sign that's more common in certain of these diseases, but the final answer is the laboratory. So, probably heard about the microcephaly epidemics that we had in 2015, 2016 in Brazil. The epicenter of the epidemic was in Recife. The first cases and most of the cases were in Recife. Recife also has, like most of the Indian cities, right, has problems with infrastructure, right, the sanitation, piped water, and it's contribute to the proliferation of the Aedes mosquitoes. So this was the work that we did with the first Zika patients, the microcephaly patients. We had 87 confirmed cases of microcephaly. And in this study, Recife has very good records, medical records. Every year, historically, Recife had, Pernambuco State, Recife is the capital, had 25 cases of microcephaly. Only August 2015, they had over 50 cases. Only August, and then the epidemic was growing. And most of the mothers, they reported Zika symptoms. So that's where we started the investigation and established the causal link between Zika virus and microcephaly, which had not been reported before. Frank Polynesia had a big outbreak before Brazil, 2013, but they did not report it. They described very well the epidemics, but only after Brazil we reported, they looked back at their data and they found they also had microcephaly. They just didn't detect it because it's a statistical thing, right? If this microcephaly is rare, 1 out of 3 women with Zika virus infection during pregnancy will have a child with microcephaly. But if you have a small population, you don't detect it, right? If you don't have good medical records, you don't detect it, right? And we had both. So these are some of the pictures from these cases. So besides microcephaly, you see arthrogryposis, these malformations of the limbs, ventriculomegaly, calcification, and also lung paralysis, the paralysis of the phrenic nerve. So it was the first time, we should not call it microcephaly, we should call Zika congenital syndrome, right? CZS, congenital Zika syndrome, because microcephaly doesn't come alone. And probably there are other aspects of disease that may show up maybe later in life, we don't know. Some psychological problems, some psychiatric problems, right? We don't know. So this is the distribution of these arboviruses, Zika, dengue, chikungunya. The red ones, like Brazil is here, and also India have the 3, right? Some countries have 1, others 2, other 3. We have 3 of them. And it overlaps with the Aedes aegypti distribution, the mosquito vector is the main form of transmission. Zika is also transmitted sexually, right? Different than Zika and chikungunya, different than dengue and chikungunya. Zika can be transmitted sexually, but the main way of transmission is through mosquitoes. And this is the situation of, if you go to outskirts of Recife, the slums, right? Big cities in Brazil, similar to India, they have, right, this type of conditions, sewage, right? No waste treatment, and people that live here, we need to think about these people, not only the neglected diseases that affect the poor, right? But also about the neglected people, because these people are exposed to several diseases and several hurdles, right? Like violence, drugs, right? They are much more exposed than other people. So, and one thing that we talk about is the, like, dengue. There's an outbreak of dengue going on, right? We work with it. COVID-19, everyone is quiet about it. But only this year in Brazil, 10 times more people have died of COVID-19 than dengue. Although we have a vaccine for COVID. So even with vaccines, we need antivirals. Because the vaccine is never 100%, right? So you have to have vaccines and treatments. So this is the effect of vaccination. If you compare unvaccinated people, the death rate, 37%, with people that take two doses of the vaccine, 0.8%. So vaccines are the main tool for fighting disease, right? But we should not forget that the vaccines and medicines, they are not the answer for everything. The most important aspects are these public health measures: potable water, adequate sewage treatment, vector control, blood and blood product testing, not sharing needles, right? Good food preparation practice, so you don't have these foodborne diseases. Biosafety, use or disposal of gloves, like in the lab. We went to visit the Ratti lab, in Dr. Ratti's company in the morning, all dress up, right? Gloves, gloves, that's very important. Safe sex condom use, if few sexual partners. This happened with the monkeypox epidemics. So people that had the disease, on average, in this cohort of patients, in several countries, Europe, Latin America, they have 5 partners in the last 3 months. It varies from 3 to 15. So many sexual partners, disease spread very quickly, right, sexually. And sexual, environmental, and health education. So, in the second part of my talk, I'll show you some of the work that we have been doing with Dr. Ratti in collaboration with antivirals. But before going that, I want to show you another area of research from our lab that we think is very important, is the diagnostic platforms. Because in order to have vaccines and good treatment, you have to properly diagnose the patient. And the diagnostic test has to be fast. In this article, it's not our article, but it's a very interesting article. They talk about test sensitivity is secondary to frequency and turnaround time for COVID screening tests. If you do a COVID-19 test, it takes 5 days to get the results, like PCR. It's useless, right? The doctor can do nothing. And you'll be around spreading the virus. So the diagnosis has to be right away, has to be point of care. And that's what we have been doing. We wanted to extend this collaboration with Dr. Ratti, develop products, because now is another stage of our collaboration, besides the scientific one. We want to have products, right? The paper is important, but the product, people need the product. So this is some of the work that we have done for Zika. This technique is called LAMP, Loop-Mediated Isothermal Amplification, is the abbreviation for it. We have published 2 papers in scientific reports, one in mosquitoes, to detect if the mosquito is infected or not, and another one with humans. I'll show you very quickly, but it's very simple. There are several ways you can do the test. Whoever is interested in it, you can go and read this review that we published. We talk about all the several ways you can do LAMP assays. There's so many different ways. We chose the simplest form of it. Very simple. No equipment required. And how we do it, we get the Eppendorf tube. We have our target. We put the reagents and your sample. Could be any sample, any biological sample. We have tested blood, serum, urine, saliva, mosquito, semen, anything. You can put in the tube and you incubate. It has an isothermal amplification. The temperature is not the same for every assay. You have to standardize that. You A lot of work has to be done for that. And then, so the enzyme that's present, it will amplify the virus genome or any DNA or RNA target that you have, can be done for anything. So if it is negative, the sample become orange. If it's positive, it become yellow. So you read the results by naked eye in 20 minutes. If it's 20, 30 minutes, you get the results done. So the advantage is very sensitive, much more sensitive than PCR, very specific, very robust technique. It is not inhibited by common PCR inhibitors. Very simple, practical, right? This can be done for undergraduate student. A technician can do the assay, rapid and low cost. It costs, we did some cost calculation. It costs, is 50 times cheaper than PCR. So this is the review I told you, and this is the real result that we have. This one assay we did for Zika, this Zika. You see Zika become yellow, and all the other controls, the non-template control, dengue from 1 to 4, yellow fever and chikungunya, some viruses are closely related, they become orange. So it's very specific for Zika. This is looking by the naked eye, the upper panel. In P panel, looking under UV light, the negative one become blue, and the Zika one become translucent, right? Transparent. When you run a gel, the same sample, you don't see amplification, and here you see several amplification of the Zika genome. So you can do the readout by different ways. We prefer the naked eye because it's simple, right? And this is the sensitivity. We did a sensitivity assay, we get the virus, we grow the virus in our lab. We are a virology lab. We grow it, and then you have it 10 to the 5, it's 100,000 PFU, 100,000 virus. We dilute it down, tenfold dilution, up to 10 to minus 7. And you can see the detection up to 10 to minus 6, you detect it. And PCR, this is the detection limit by PCR, can only detect 10 copies. So our assay in this experiment was 1,000,000 times more sensitive than PCR. But just one experiment. You have to do it 10 times and do a probit calculation to be fair, right? So what's the detection limit? And 95% of the times. So when you did that 10 times, the sensitivity decreased a little, but it's 10,000 times more sensitive than PCR. It can be up to 1,000,000, but not 95% of the times. It's going to be 1,000,000 times more sensitive, 20% of the times. So you have to do several repetitions, but it's much more sensitive than PCR. At this, we did a validation with Zika patients, those patients from the epidemics we had in Brazil, we had the samples. So this is the threshold for PCR, which is 38, the standard test, right? The ones in the left are positive, the ones on the right are negative. So you see that some of the negative samples by PCR, they become positive by lump, because it's more sensitive, right? And it's cheaper. So besides having good diagnostics, you have to have a diagnostic to treat the patients. We need to develop antivirus. And antivirus, whoever have read about, have studied microbiology or pharmacology, you learn about the antibiotics, right? The medicine that we have available. Lots of them are antibiotics. For viruses, we have very few antivirus in the market. Over 6 years of research, we only have about 100 antivirus approved for human use. And most of them are for chronic viral infection, like HIV, hepatitis B, hepatitis C, herpes virus, right? Why is that? Because infection is chronic throughout your life. You have many opportunities to treat the patient. An acute infection like Zika, dengue, the new coronavirus, the virus will replicate in your body for 1-2 weeks, it will be gone. So we don't have time for the antivirus to work. So viral infection is very acute, it can be very acute, depending on the viral infection. That's why most of the antivirals developed are for chronic viral infections. So, why there are so few antivirals? Different bacteria, which are prokaryotes, viruses use the host cell machinery. They use the ribosomes. They use all the enzymes, the proteases. So if you block the virus, you're going to interfere with the cell metabolism. You're going to have side effects. So they're not going to be safe. It's very difficult to find a molecule that's safe for the cells and can kill the virus. So this is a challenge to develop. And some of the viruses are very difficult to develop antivirus. You cannot grow them in culture. Like HPV, you cannot grow the virus. How can you test an antivirus? It's very difficult. Norovirus, HPV. Some viruses are too dangerous in smallpox. Or like in India, you guys had an outbreak this year of Nipah virus. It's a BSL-4 virus. Which lab in India can test a drug under BSL-2 conditions, right? Very few. Some viruses don't have good animal models, like smallpox. You cannot test in vivo. Only humans are the host. This is very challenging. And a compound, it must block viral replication very efficiently. Like this one, the optimal dose. Because if you have intermediate dose or low dose, the virus will mutate. They are mutating all the time. And the drug will select the mutant, which have resistance to the drug. So the drug will be useless. So, another thing that I told you, most infections are short-lived, like the acute infection. The virus stays for a very short period in your body, different than the persistent infection, like herpes virus or HIV, that stay throughout your life. So for this acute infection, it's very difficult to have a very short window of time to treat the patient. And you should not be giving antivirals to people at random, right? You have to test the patients. Very few exceptions can happen, like the PrEP, for people that are HIV positive. It's recommended they take the antivirals before having sexual contact with other HIV infected person, non-HIV. But for most viral infections, we don't do pre-treatment. And they are not broad-spectrum antivirals because each virus is specific. If you test an antiviral for dengue, it doesn't mean it's going to work for Zika, although the viruses are closely related, because viruses are very specific. Zika is not dengue. Dengue doesn't cause microcephaly. Zika does. So the viruses are different. And the pathway to antiviral discovery is a long pathway. It takes 5 to 10 years, right? It's very expensive. It can cost 100, 500 million US dollars. And if we start from 100,000 compounds, we will finish with one FDA approved compound. So most compounds, 1 to 100,000 on average. So it's very difficult to have an antiviral on the market. Most of them will fail. But it's not because of that we are going to give up, right? We are continuously working on antiviral discovery and development. I'll show you some of the work that we have done. So we look for antivirals, we search for antivirals from natural sources. For that, we partner with chemists, pharmacists, they isolate these compounds, sent to our lab. This is a work that we did, use the geopropositories from Brazilian bee. It's called the Jandaíra bee. It only happens in the northeast of Brazil. It's an area similar to Rajasthan, like a semi-arid area, like a desert-like area. These bees grow in the forest of these areas. So very complex propositories and honey. We isolate the compounds, and these are the two compounds that they collaborate, naringenin and 7-methyl-naringenin. We tested, Poliana, my PhD student, she tested for Zika and for dengue. And this is a publication, not going through the details, right, of the papers, but have very good activity against both Zika and dengue. This is the reduction. We have different fractions of the compound, the ethanolic, acetethanolic, oxalic fraction. So you see the compared to the controls, we have a dose-dependent effect, and these are the isolated compounds, because for antiviral discovery and for the publications, the reviewers always ask for the isolated compound. You can test the fraction, but you must have the isolated compound. These are the two isolated compounds we tested. And now we are doing some encapsulation. We are using nanotechnology to encapsulate these drugs so it can cross better the blood-brain barrier. Thank you, sir. Because for Zika, we need this compound to reach the brain, right? It has to cross the blood-brain barrier. So Poliana also, during her master's, she did another work from Brazilian plant. This plant, the native Brazilians, they use these plants for lung problems and some from fever-related diseases for several uses. So we got this plant, we isolated two compounds, two bis-benzyl quinolone compounds, varifitein, methylvarifitein, and test against Zika and dengue, and they had very good activity against both viruses. This is another work we did in collaboration with Carol, Ana Carolina, it's a professor, a very good virologist from Brazil, and we helped her with Zika assay. She isolates some peptides from Brazilian snakes. There are only 4 snakes, 4 gender of snakes in Brazil that are poisonous, different than India. So you have to have the anti-serum specific against these snakes. If you use an anti-serum used in Africa, India, Europe, it will not work in Brazil. It has to be specific against the Brazilian snakes. But it has some antiviral property. So you never know. Serendipity, right? You have to test it and see if it works or not. So this is the work we did in collaboration with Ronaldo, which is here, and also Rati. These are the guys that understand chemistry. I know not about, I would fail any chemistry exam that you give to me, but they are very good. They send us the chemicals. They teach us how to solubilize these chemicals. It's very important. So give the compounds, teach us how to solubilize it, we'll test it for you, right? So we did that for Zika. And had very good activity against Zika. This is Williane who works a PhD student of Ronaldo and I. She's in my lab. She's finished, gonna finish this year. We test the same, similar compounds, another series of compounds for chikungunya. These are undergraduate students. In Brazil, the undergraduate students have a nice program. We call the scientific initiation. They come to our lab, there's a selection process. They start to get trained in science. They stay 20 hours a week in the lab. They make 500 reais, it's going to be 5,000, like 80,000 rupees a month. It's good, right? It's good, right? 8,000 rupees a month. They say for 1 year, doing research. This is Melissa. She's been 6 months in my lab. She's very close to get a scientific publication in a patent. She's testing compounds against chikungunya. Chikungunya, these are the various plaques. It's a plaque assay. When you ladies go to Brazil, the two that are going, we'll teach you how to do this. It's my favorite technique in virology, the plaque assay. You know, you don't see the virus, but you see the effect of the virus. Each plaque has thousands of viruses, but came from a single virus. So then you can compare your compound with your controls, if it's killing the virus or not, right? You love this assay. It's a Nobel Prize. 1950, tobacco, he developed the system. So he got a Nobel Prize for that. Without this assay, we wouldn't have the vaccines against polio, right? The saving Salk vaccines, or against any diseases. Because we need to grow the virus. If you don't know how to grow the virus, how can you make a vaccine? How can you test it, right? How can you have an antiviral? And this is the work that we have done several molecules of Rati. He's very productive. He sent us lots of molecules. He makes us work a lot, right? But we love him because very fast, right? Very reliable. He can tell you all the structure. I know nothing. But this was just accepted. And RSC, medical chemistry. And my PhD student, Renata, she's the one leading these studies in Brazil, the antiviral. There's many students in RT groups synthesizing, doing all the chemistry, the in silico work. But it has very good activity against the SARS-CoV-2, the new coronavirus. We work in the BSL-3 lab, right? The work is slower, takes more time, very dangerous, you have to be very careful. Zero tolerance for mistakes. Before each step, you have to think, because there is no mistake allowed, right? BSL-3, you cannot be working in the lab like very comfortably, right? Without thinking much. Before you do each movement, you need to think. But we train people, right? We train, they are very well trained, no problem. So these are the negative controls. You see the virus destroying the cells, very aggressive viral infection. The mock, non-infected cells, look healthy. And you try different concentration of the compounds, the three best ones, 3G, 3I, 4D. This is CC20, is a critical concentration for 20% of the cells. We don't test the compounds at the same concentration. We can do that, there are several ways of doing. We prefer that. In the screening, we test which is the concentration that is safe for 80% of the cells, which means it kills 20% of the cells. This is the maximum that we use. Our compounds are tested at the same concentration, they say C20, so give the same chance to different compounds, right? If they are very active, they're going to work well against the virus, right? And then we do it blindly with the controls. So these three had the best activities. So antiviral discovery development is an exciting endless task because viruses will always come to infect humans and animals, and they always mutate against the antivirus that we already have. So we need to develop new antivirals. So with that, I'd like to thank your attention all for coming here, for hosting us so nicely. These are the people from my lab that do the work. I also would like to thank our collaborator, Dr. Rati, Dr. Ronaldo. So this 2019, 2024, we met again, and we will meet again. So it has been very pleasant. We are very happy to be with each other, to work with each other. And I was preparing for this talk, and I was checking how many papers we got. We got 7 papers, Hachi. That's a lot, right? In 3 years. And we have another one that just accepted. So, thank you very much for your attention.
A very good afternoon to all my respected colleagues and dear students. It's my privilege and honor to welcome our next speaker, Professor Ronaldo Oliveira. Professor Ronaldo is a professor. He's working at Federal Rural University of Pernambuco. Okay. At Federal Rural University of Pernambuco, and is a chemist and whose main focus is organic synthesis, heterocycles, and carbohydrate chemistry. And his research interest includes design, synthesis, and biological evaluation of small molecules. Professor Ronaldo has supervised a PhD thesis of 8 students, 13 master's students, and 1 postdoctoral student, and he has numerous publications and has 5 patents to his credit. So without taking any more time, I would like to invite Professor Ronaldo and share his research with us. Thank you.
So thank you for the introduction. In fact, I knew Professor Ratti by mail, 2017, I think. We try new breeds, malaria, but this not work. Now it work with bricks COVID-19. So sometimes we apply, no results. So you need work, hard work. So you have results after that. So today I will focus on some work searching bioactive compound against viral disease. So the last 10 years I work with these diseases, cancer, tuberculosis, malaria, Chagas disease, nutrição, preparação cruz, leishmania, and anti-inflammatory. Today I will focus just Zika virus and SARS-CoV-2 results between collaboration between INDO-BRAZILIan. We started this in 2015 when we have the problem with Zika virus. So I like to work with carbohydrate chemistry. So it's not very easy, even for students, because you need to protect the protector reaction. It's very hard. Purification all the time. It's very difficult. Some crystallization in water. So it's very, very, very hard. But we know a lot of things about the chemical reaction because the major of reaction don't work. So you try 1 time, 2 times, 3 times, a lot of conditions to arrive good results. So my strategy is to work also with some heterocycles on more sample reaction. And with this strategy, diversity of compounds, synthesis, and also diversity collaboration with other colleagues in biologists area. So with this strategy, nowadays I see that I have a lot of bunch of results. So these are my research panel for today. Okay. You can see in middle simple molecules like glucose, naphthalene, glycerol carbonate that you can prepare this from glycerol. This is byproduct from biodiesel preparation. And also some nucleobases. So With this, you can see the first step in green is a glycosylation reaction. After that, you can open the carbonate and prepare the rest of compounds. On the other hand, in red, you can prepare this derivative. It seems some acyclic compounds based on nuclear bases and these carbonates, glycerol carbonate. We can start some scaffold based on sugar. So here's just some background about lactobacillus. We have a lot of reasons, good reasons for activity, Chagas disease, leishmania, cancer. The problem with this structure with lactobacillus is that they're high cytotoxic. So when you have lactobacillus as a scaffold, you have toxic molecules. So the goal is became these molecules less toxic. We arrived this when we prepare the amino lactobacillus. The triazole directly conjugates with lactobacillus is toxic. But when you have amino lactobacillus, you have less toxic compounds. So this was a good reasons, very nice reasons. For instance, we have this strategy for anti-malarial compounds. You can see up the strategies use azide to prepare the amino lactobacillus and azide and triazole at the end, final compounds. And the other strategy, you have the compounds triazole also, but you can see we invert the triazole connection, the acyl connection with amino lactobacillus. On the other hand, propagyl connection with amino lactobacillus. So the up compound is better for malaria, against malaria. See, you can see carbohydrate-based sugar lunches during the last 20 years. All the structure of based antiviral, based on the nucleosides. You can see the structure, right? And my focus is not furanoside structure, but pyranoside structure. So it's more easy because you can start from glucose, for instance, less sugar. So it's very nice case. You can see about the last year, because the pandemic situation, we have a lot of introduction to carbohydrate, luncheon, structure as antivirals. So we are motivated. In 2015, with Professor Inomar to prepare a lot of compounds because this problem is Zika virus in Recife. So all these compounds around was more or less good for Zika virus, against Zika virus, but the best was this ground example of compounds. We can see phthalmide, triazole, sugar, moieties. So this strategy today is to prepare all compounds by glycosylation reaction and heat chemistry. This result that I will show today started in 2016 with this reaction. You can see it started from the glucose and glycerol, these renewable resources. So after glycosylation reaction, like the 10-year reaction, and the best result, very interesting result, was in this step, where we found the compound 3 as on poor diastereoisome. How this make it? just a resolution by crystallization. You have, if you see this reaction, you can reach 4 compounds, alpha, beta, and L as configuration in agricole portion. So after crystallization, 30% of the yields, you have this compound, alpha, derivative, and the S configuration in agricole portion. It's a very good derivative. You can compare with like faster resolution, spontaneous resolution by crystallization. You can see carbohydrate is not easy. All the time you need purification. We just one purification sample, and after that, crystallization. This is very difficult. I think so. We have chance with the results. So after that, we started a lot of derivatives. We prepared a lot of derivatives. These derivatives that show me, I know I will not show for you, have had a very good results against tuberculosis. Very nice. I will not show it today. In this new compound, also a lot of derivatives. You can see the nucleobases connected to this compound from glycerol carbonate linked to sugar. You have nice results, less than 10 micromolar gas ether, some molecules. So this 10 micromolar is a line for we try to find new compounds or leader compound, because more than this is not good, not very good. But less 10 micromolar is a very nice result for the biologists continue the study and in vivo testing, for instance. These results also you can see the same scaffold, sugar unsaturated, glycerol portion, triazole, and now natrophenol. To have very good results against antithyrosinase and against melanoma. The question is, maybe this compound have some activity against virus. I don't know. Of course, this is not a rationalization thing, but it's more serendipity or by chance to find the new antiviral compounds. So the synthesis, first step I show, Ferrier reaction, you use the catalyst, the new catalyst. You published this 2015. And after open the glycerol portion, the carbonate portion, cyclic carbonate portion, you have this azide, so this is precursor very, very easy to prepare. Maybe you need the protection steps. So after you have this bunch of molecules, you can see the products, and the result is very, very good. But even we contain naphtophenol derivatives, aminonaphtophenol. You can see agasterosinase, 4 micromolar, agaste cancer, melanoma. Nice results if you compare with dots. The result is very good. So the question is, this compound may be antiviral results. You can see another leader compound without naphtophenol in this structure. So this is a nice leader compound. Of course, you still change the structure. So maybe you have, in this case, a dual activity against cancer and against virus. The other results, we start this sequence of reaction. You can see it's good. You can see the results is not good selective for alpha beta compounds, and you have a problem with the alcohol triazole naphthoquinone because you have a quench with the catalyst. So we change the sequence of reaction. You can see starting from glycoside, all glycoside, and after the click reaction. So you have a good selectivity. But still you have the problem, this compound became toxic because you have triazole naphthoquinone. So the next step, we change naphthoquinone, that you know this naphthoquinone is toxic. You change it by phytalimide. So now you can see the next step with biological activity. The phytalimide in general is not toxic. So the good results, you can see these cytotoxic results against FPG2 cells, good cytotoxic neuroblastoma cells, and vero cells. So the screening, the first screening with this compound, all compound was active against Zika virus. The in vitro results, the best result you can see this compound. We show the structure. So in this case, you can see 4 compounds, 4 leader compounds. So at now, you don't know the mechanism exactly, but you know that this molecule, the molecules work in replication step, no competition in antivirus. So we need to know now the mechanism using some enzyme to know the inhibitory concentration. Maybe this enzyme NS5, NS3. So we figure why not test for SARS-CoV-2, this kind of compound, and you have nice, good results. You can see this leader compound from the same series. So the conclusion is you have a bunch of molecules with against SARS-CoV-2 and Zika, good results. Other structure against only Zika until now. Maybe good also for SARS-CoV-2. So finally, I thank you very much for your attention, and I thanks also BRICS project. CAPS and FACEP is fellowship for the students. The students need fellowships, no? It's good for the students. If not, the exclusive education is very hard. You know? So, all the time you submit project for taking money for the students, and for fellowship for the students. So this is by the students that we work this time. My collaboration with Professor Arnaud in France, University of London. You know this guy, Lindomar. He's the big guy, the very nice guy, Latino, Celso. This is my research group at this moment. And finally, I invite you to Brazil, maybe the next conference. To convince you, my view, my series is just Eastern Orient receive. So, convince you to go to Brazil. So, the life is not work, not only work. Sometimes go to the beach. I show this for you. And finally, the last photos. You know this guy? That's in 2009. He received. So,
first of all, I would like to thank both of our speakers for such an informative presentation. And also, I would like to mention that the presentation was so much easy, precise, and it was presented in a very in such a manner that it was understandable for like each of us understood it very clearly. Now there must be many questions in our young minds, so the house is open for questions. So if anyone wants to ask any question.
So I have questions for Dr. Lindomar. Um, First of all, the presentation was really clear, crispy, and actually informative. So in your LAMP essay, you said that this is qualitative, and why not quantitative? Because color gradations can be seen in, like we do in ELISA and other things, so we can quantitate it as well. So, yes.
That's a very good question. We do it qualitatively, yes or no, right?
Yes.
So that's the most important information that the doctor needs
Yes.
the patient needs,
Yes.
right? So, but the quantity, the viral load is also very important for you to predict how severe is that case.
Yes,
Because
yes.
one thing is if you are shedding, or if you have, let's say, 1,000 virus in your
Yes.
nasal circulation. Another thing is if you have 1,000,000 virus per ml in nasal circulation, right? It can be done quantitatively, but that you need an instrument for that. You have to develop an instrument or buy one because it's a color change, right?
Yes.
You have to measure the change in
Yes.
color. You can do it even in real time. People have done it.
Yes.
But the thing that we prefer not that system because we want to keep it simple. You know, you can take it to very remote areas, no sophistication, no equipment, just a water bath. You can do it.
Okay, so means it can be done.
It can be done.
qualitatively.
It can be
Okay.
done. Yes.
So another question. I am full of questions right now. So you said that it is very fast as compared to PCR. But what about its efficiency? Like you said, you have to repeat the test for this. But how is it efficient, or is the efficiency compromised, and we have to repeat it several times?
It's faster than PCR. PCR, the assay varies, but
Yes.
on average 3-4 hours, right?
Yes.
Running time.
Yes. Yes.
That assay takes 30 minutes.
Yes.
30 minutes to have the results. So we do it in a test tube
Yeah.
because we don't process thousands of samples a day,
Yes.
right? But that can be done. It's just you can develop the same assay in a 96 well plate format,
Yes.
or even 384 plate, right? You can
Yes.
save reagents, you can make it cheaper and faster. So there are different ways of doing it. We opted to have it the simplest way possible. Happened of two, very cheap, water bath, right? No equipment. That's how we did it. But there are other
Yes.
ways you can do it.
But efficiency, is it like, is it successful in detection? It detect the diagnose the disease 100%, or efficiency is compromised in this case?
No compromise. It's much more sensitive than PCR.
More sensitive.
It's as specific as PCR. The only thing you have to be careful with this assay because it's too sensitive. It's very sensitive. Right? Sometimes it can become too sensitive, right?
Yes.
So you have to work in a very clean conditions, right? We work in the bench because at the beginning, I had a postdoctoral student, and this person, I hired him. He was working like in a BSL-3 lab, all the gear. I said, forget about it. He was not working in the virology lab. He was working in the parasitology lab because there was no Zika. I said, no. If you get the results are not true, right? You have to have the test work in a Zika infected lab, Zika contaminated lab. If it doesn't work, it doesn't work, right? You're not going to publish anything that's not good, right? So forget about it. Do it in the bench, right? And then he stayed 1 year, but it was very difficult. There's not nothing more motivated. Then I had a master's student, first year master's student. I called Jefferson. He was my undergraduate student. He's a very good person, very good scientist. I called him in my office and said, Jefferson, stay with him, right? Because I think I'm gonna need your help because I have to have accountability. Because I got the money, right? I have to get the results. So the postdoc stayed 1 year, 0 results. Jefferson, 3 months. We got all the results. He got 8 prizes
Wow.
in scientific meetings. And then he did a master's, PhD. He got 15 papers in my lab. And then the third year of the PhD, a collaborator of mine from Canada asked me if he could offer him a position to his lab So he took him to Canada. He's in Canada now doing a postdoc.
Great.
Yeah, so they write the most important element in the chemical table is the H2 element, the human element, right? When you add the human element, the chemistry changes, everything changes. You can have beautiful labs, right? Very good infrastructure. You have loads of money, research money, but you have to have the good people makes all the difference.
So it's a message for all our undergrad students. There's a good scope. His undergrad students work really hard to get publications in 3 months.
Yeah.
So you can also do that. Last question. In your slides, you have shown you have extracted antivirals from poisonous animals, like let it be bees, snakes. Why? What is so specific in poisonous animals that you have picked snake, bees, and why not phytocompounds or maybe some other animals which are easy to handle?
That's another very good point. The thing about the drug discovery, right? You never know. So where you find it, you only know if you test it. The prediction is very good, right? To do the rational drug design, but I myself prefer to do it after. I know it works. There's sometimes Ratty, remember? Ratty sent us some compounds. We tested. It didn't work very well in the first screening, and then Ratty, well, test this compound again because we did some prediction in silico. This one must work, right? And I asked the student, try these other compounds blindly, and the other Hachi told us that in silico it worked. So I think it works both ways, right? You can do it randomly by serendipity, right? Just testing. But sometimes also by computer-based design, right? I think both approaches they are complementary. Even though these animals are poisonous, this Brazilian bee is not poisonous. It doesn't bite you.
Okay.
But the honey is very expensive because it's very easy to handle, but produce very little. But the poison snake is very poisonous,
Yes.
right? But it's not the whole venom. The venom is very complex. You have thousands of substances. It's just one peptide. So among all the toxic compounds, there's this one that is safe. It can be used. Actually, there is a capopil was isolated from Brazilian snake.
Okay.
That's how Brazil became very protected of its biodiversity, because some companies from developed countries could go to Brazil, to the Amazon, and harvest the biodiversity, right? And then give nothing back to the country. So now all the biodiversity is very well protected by laws. You cannot take like a Brazilian snake, a plant, and just take to a foreign country without the proper paperwork. Yeah.
Thank you so much. Thank you.
Thank you.
Once again, thank you, sir, and thank you, audience, for your engaging participation. Now I would like to call upon Dr. Lokesh Chandra Misra, sir, to present a token of appreciation to Dr. Lindomar Pena. With a huge round of applause. And now I request Dr. Shailendra Kumar, sir, teacher in charge, Department of Chemistry, to present a token of appreciation to Professor Ronaldo.